Okay, thank you. I'm so delighted to be here. I'll make a little bit more formal statement of thanks to Dr. Stephen Jones and to Kelly and to everyone who made my visit possible. I've been wanting to come up here to the Skagit Valley and to the research station for a long time, since I had the great honor of having Dr. Jones be a keynote speaker at a conference I organized back in 2011 at the University of Oregon when I was a professor there. So what I want to do today is talk to you about an area of my own research and a kind of area of experimental art that I hope will be thought-provoking in terms of this very diverse and sometimes conflicting set of ideas about what would constitute a sustainable food system in the future. And also to kind of provoke us to think deeply about some of the relationships between art, science, and practice that are happening out there around food and agriculture. So I'll begin by actually offering a kind of overview of this area of art known as wet biology art, or for short, bioart. Most of the artists who fall under this rubric, this heading, are themselves polymaths. They're trained in the visual arts, in design and media arts, but they're also often trained in another or multiple other disciplines: computer science and computer programming, product design, biology of one discipline or another, molecular, cell, etc. And they bring this sort of sense of art as having always been transdisciplinary, having always been that which deploys the imagination to engage with forms of knowledge that are in process and that are unfolding. They are these sort of Renaissance men and women, to use the kind of lay term. So before I kind of get to a set of bioartists who are really provoking us to think about the future of food, I wanted to offer this kind of wider overview. And at the outset, I should say that I welcome you to jump in and chime in with questions or comments at any point. I'll stop a few points along the way, and I think we'll have some time for conversation at the end, but please just interrupt me at any point you feel inspired to do so. And I really welcome conversation and debate about some of the issues these artists are engaging with. So what is bioart? I'm curious how many folks have heard the term before. Not a one. And I think that suggests something, which is that one of the challenges for art in the 21st century is to get out of its silo of the gallery and the museum and out of a kind of elite group of people who follow experimental art. Contemporary art has, since the Second World War, had this kind of challenge because of the way that institutions of art, art celebrity and art sort of authority are increasingly shut off from the vast majority of the public. But in fact, even though you haven't heard the term bioart, these are artists who care very deeply about getting out of the gallery and out of the museum and actually creating new public forums for us to think deeply about some of our most complex issues around biological technology in particular. So in my research, what I've identified are several different forms that bioart tends to take, and I have a few of these outlined on this slide. So the first of these is the kind of cultivation, if you will, of transgenic organisms in gallery settings. So what certain bioartists do is they collaborate often with molecular biologists to create novel life forms and to use transgenesis in order to create transgenic organisms that are not serving a utilitarian function. They are not meant to be planted in fields or meant to be used in medical research or medical science. They are sort of thought experiments, if you will. And I'll give you an example of this in a moment. But the other thing that is a consequence of this introduction of a transgenic organism into a gallery setting, into an art setting, is a much more philosophical inquiry into the boundaries between art and science in the modern era. And to my prior point, the sort of reminder to us that those boundaries were not always so rigid. There wasn't always such a separation between art and science. And we now have almost codified that separation through laws, as you'll hear a bit more about. A sort of subset, but it doesn't fall fully in the prior category, is a group of bioartists who are trying to create novel foods. And these two have a kind of non-practical side. They are about thought experiments as well. And then many of those bioartists take another step, and they put these novel foods, and I'll tell you about a couple, into a performance. They engage a public to actually eat, or at least be disgusted by the thought of eating these novel foods. So let me give you one example, because it's at this intersection of those two middle bullet points, if you will, that my research is focusing right now. So there's a group called the Center for Genomic Gastronomy, headed up by Zach Denfeld and Kat Kramer, artists by training, but also well-versed in molecular biology, cell biology, and computer programming. And they have basically been working with a team of scientists on in vitro meat, which, as some of you may know, is an area of scientific research that many serious scientific labs have undertaken to pursue. And so there are labs in Europe and the United States and elsewhere where there are teams of scientists really focused on trying to figure out whether we could culture meat in vitro as a sustainable source of animal protein in the future. There are all sorts of technical challenges that these scientists have encountered, about which we can discuss. But Zach Denfeld and Kat Kramer of the Center for Genomic Gastronomy are less interested in whether this may be a long-term viable alternative to raising animals in either feedlots or on ranches. They want to provoke us to explore the boundaries between plant and animal, and our sort of sense of what kinds of foods we're willing to have manipulated in these ways or cultured in these ways, and which we're not. So they staged a kind of Iron Chef style competition. This was at a very famous sort of art exhibit and art festival that happens every year in Rotterdam in the Netherlands, called the Electronic Arts Festival. And so I was in attendance. And there were the room was set up just as Iron Chef, if you can picture that. So there were two teams, two kitchens, and there were all sorts of ingredients that we might expect to find. There were tomatoes and parsley and shallot and garlic, and there were condiments, olive oil, and so forth. So it looked as if, what you would expect to encounter in a kitchen. And then there were two vats of in vitro meat, very small. Actually, they were test tubes, vials really, of in vitro meat that had been cultured in their collaboration with these scientists. And that was, if you know the show Iron Chef, that was the challenge ingredient to work with for the evening, was the in vitro meat. And then at the end, there was a tasting. Right? And only a very small number of the 200 or 300 people in the audience took them up on this tasting. So I want you just to hold this in your mind as I sort of offer some other ways of thinking about experiments like this and what's at stake. And you may be very skeptical that this kind of performance is anything more than just a kind of intellectual exercise or something that is too abstract to really make a difference in how we think about the food system. But this gives you a kind of example of where some of these artists are taking their interest in food and in food production. And I use this term happenings with a very particular sense in mind. Happenings was the term used in the 1960s to describe a group of very radical artists who were completely abandoning the gallery and the museum altogether, rejecting those spaces as too elite, too out of touch, and they started to stage all of these performances in everyday places. And one of the most famous of those that actually touches on food politics happened in Berkeley, in People's Park, a group called the Robin Hood Commission, something like that. It sounds like you maybe remember this protest. Let's see, the exact nature of the happening was they arrived and they started to illegally install picnic tables and plant fruit trees and sort of recreate this park as actual sort of community garden, which may sound very familiar to us today in 2013, but was a very radical act. And it was really art and activism meeting with horticulture, in fact. So finally, and this will be my last example today, so I won't preview it now, there's a group of these bioartists who are interested in using things like in vitro meat as a building material. And so I will come back to that example and what is going on with that kind of bioart. Okay, so for my part, rather than kind of focusing on what bioart contributes to the wider contemporary art world, I'm really looking at the question of what they are offering to the future of environmentalism and specifically to the future of food sustainability. What are some of the new values and paradigms they're offering up for us to at least consider, if not accept? But before getting to food per se, I want to show you the most famous bioartist and an example of one of his most notorious projects, Eduardo Katz, who created this installation, Genesis. And the thing to know about Eduardo Katz is he is very much of the elite art world. A renowned artist. He's exhibited in all of the major museums of contemporary art, as well as in many very elite gallery settings. In 2000, he created this project called Genesis. He collaborated with a team of molecular biologists, and in short, what they did was took the bacteria it is E. coli, right? Yes, there are a lot of different bacteria that come up in bioart, so I always want to make sure I'm getting it right. So he took the bacteria E. coli, and with these scientists, they used a plasmid molecule to correct me if I'm getting any of the technical details wrong, in order to insert a fluorescent marker so that they would glow. Right? And of course, as many of you know, this kind of glow-in-the-dark form of genetic engineering has become now a really commercially profitable or lucrative form of genetic engineering. The glowfish, most famously, these aquarium fish who glow in the dark. So this was prior to the patenting of the glowfish, I should note. So then these genetically modified bacteria were installed under these UV lights, and the UV lights were then producing further genetic mutations. But participants could also manipulate the lights, the intensity or whether they were on or off, as could people over a web channel. So people not physically there could also participate in this sort of active mutation or sort of genetic engineering. So there's more to it than that, though, because what Katz and the medical or the scientist team had done is had translated Genesis 1.26, right, book 1, verse 26, into Morse code. That's the famous lines from Genesis about dominion over fish and sea, does anyone know the exact verse? I didn't write it down. No, okay. Oh, wait, let man have dominion over the fish of the sea and over the fowl of the air and over everything living that moves upon the earth. So using a kind of conversion principle, they converted that into a DNA sequence where A, T, G, and C were used to translate that book of. So what happened at the end is that the new genetic sequence after all of the mutations was translated back into standard English through this same conversion principle. And it was, of course, nonsense. So this is where the kind of interest in what genetic engineering can do biologically meets up with the capacity for language and for artistic expression more generally to be another form of sort of mutation, if you will, of transformation. And in some ways, what he was doing was staging a kind of collision between these two different forms of change or of creativity, the scientific and the artistic. But at the end of the day, this, I think, is a really intellectual, erudite, abstract kind of project. And it was hard for most viewers to really understand what was at stake. Was this a critique of genetic engineering? It felt more like a kind of virtuosic performance of both his capacity to work with scientists and his capacity to sort of poke fun at genetic engineering. In addition to that, despite Katz's really big claims that Genesis represented an unprecedented collaboration between artists and scientists in the contemporary period, he gives those scientists almost no credit by name for the work they do. And so when you go to his website, what you see is Eduardo Katz, right, the artist genius. And in footnotes at the very bottom of the project page for Genesis are the scientists, both the sort of faculty and postdoctoral scholars in the lab who did the work, right, who did the labor. And so I think this actually raises some really important questions about when artists and scientists collaborate, what is it collaboration? What are the sort of structures of authority and of credit that get set into motion? But in any case, Genesis remains one of the more famous bioart projects, and it was indeed Eduardo Katz who coined the term bioart back in 1997. So this is a book, I highly recommend it if you haven't encountered it, by David Edwards called The Lab. And I wanted to pause at this point on The Lab, Creativity and Culture, with the bread lab particularly in mind here, right? Because as Stephen was showing me the bread lab and we stood at the end and looked at one side at the test tubes and the scientific equipment and on the other side at all of the culinary equipment, it sort of really drove home why this was on my mind when I was preparing for this talk, which is to say that Edwards argues that we are in a time when, on the one hand, there's been this separation of different forms of knowledge. And on the other hand, we're seeing a lot of energy, not just in the world of art, but in the world of science, to break down those barriers that have been erected since, especially since the Cold War. So in my view, the most exciting bioartists, so I've sort of offered my criticism of cats, are those that are really seeing their task as being to collaborate, to truly collaborate with scientists. And in doing so, to build new kinds of knowledge and new sorts of bridges between the arts and the sciences. There's something else, though, that they're doing in sort of breaking down the science-art division and doing it through food, which we should remember has always been both alchemical and scientific, as a practice. There's both the sort of magic of cooking and of farming, for that matter, and its technological and technical dimensions. But in addition to sort of breaking down art and science, they also are really breaking down what has been a long-standing divide between environmentalism and technological innovation. So that's what I want to now focus on. But let me just pause at this point. Are there questions or comments I can field, or things that you want to know more or less about as I sort of move into the even more food and environment-centered part of my remarks?
Do you think, in the same way that perhaps these bioartists are trying to bring their work to the public, there would be a benefit of bringing their work to the attention of scientists?
Yes.
The fact that none of us have heard of bioart before,
Right.
There's a great communication back to the scientists that
Yeah.
what has been used, other than those who directly collaborate.
Right, other than those that they've reached out to, that they have formed these connections with, it sort of remains this kind of coterie of these teams that they've worked with in the past. And there are certain labs. For example, there's a lab of wet biology art in Perth, Australia, where there are these teams of molecular and cell biologists and soil scientists and artists, computer programmers, these very multidisciplinary teams working on some of these experiments around the future of food. But I think they remain unknown to their various compatriots in the art world as well as in the science world. So yeah, I mean, I actually think this is the biggest task, is not only to find public venues for engaging the public in provocative sorts of conversations and experiments, but to engaging a wider group of the scientific community. Yeah. I mean, very few people, I'm doing area research that very few people are doing research on, which is scary. It's like, because even most humanities scholars working on contemporary art look at me blankly when I talk about bioart. So, but one of my arguments is that we should be paying attention to these people, and they also need to reach out to a broader group. Yeah.
But you, in fact, are bridging that gap yourself, right? From the workshop you had.
Right.
Study women. So I spoke right after an artist spoke.
Yeah. I paired Stephen with an artist who would not call himself a bioartist, but in my view, he fits the bill. He and his two collaborators in Los Angeles are part of Fallen Fruit, which got some coverage in the New York Times. They basically they create these hand-drawn maps of fruit that's in the public domain, whether it's a branch hanging over into the public domain or an entire fruit tree. So they map neighborhoods to show people where they can access public fruit. And then they also helped do a kind of art installation, but it was a park, a public fruit park in a part of Los Angeles that they're trying to use as a template for other parks. And they often are collaborating with both policymakers and scientists. And so it was really exciting to have the two of you speak back to back like that. So I think the more we can have those kinds of conversations, it's difficult because you're speaking different languages on the surface, but really only on the surface. Yeah.
Sorry to hop on.
Yeah, no.
What about in schools? I mean, kids love to draw and
Yeah.
art, and they tend to find science boring. And this would be a wonderful way to move
Right.
to a creative, engaging
Yeah.
form of teaching.
That's a great idea. I think actually to form a kind of bioart curriculum for K through 12 would be a really exciting undertaking. Yeah, I haven't thought about that component. I mean, I will say that my environmental studies students light up about this once they start to dig in. It's like, oh, this is a place where I can bring these really disparate, because a lot of environmental studies majors, they have interest in storytelling, but they also have interest in the hard sciences, and how do we make these meet? Yeah, so.
What's the limits to how far you can go for that science?
Yes. Yeah, I mean, what occurs to you as being one of the limits?
They're quite different ways of using the brain.
Yeah.
It's quite important that they're different.
Right.
Art is for imagination and like it's a playful thing. Science doesn't necessarily work that well if you're just trying to be free and playful.
Free and playful. Yeah, I think you're right. I think there's in trying to imagine a less divided structure in which art happens in one realm and science in a completely other realm, and the study of art, has nothing to say to scientists, which to me is an even more fundamental problem, actually. I think in thinking about collaboration, it's not to suggest you make murky and muddy and you just sort of mix all of the skills into one big vat, but rather you really leverage these different ways of producing knowledge and of understanding the world, including, and actually to have two people in a room or multiple people from different sides of this in a room where data is one, data defined sort of broadly, data is one form of knowledge and the imagination is another. And I think that the contact, rather than the sort of collapsing of those boundaries, the contact between them, I think, is productive. But I think you're right. And also there's the kind of workaday science that just requires an incredible amount of time and detailed, sort of analysis. And the one thing about some of these collaborations, as I know about, is it can distract. I mean, it pulls a lot of attention, right, away from what may be the main research question or hypothesis. I mean, artists don't really work around the hypothesis model, the scientific method. I mean, they're interested, they're the provocateurs, right? So, no, I think you're right. There are questions asked about how these collaborations maintain some of the integrity on each side of what the techniques are, what the methods are, and also what the sort of social function is of each. Yeah.
I guess sort of building on that, you talked about artists sort of reaching out to scientists. Are you aware of scientists reaching out to artists? Like, we're doing this project,
Yeah.
It's very interesting.
Right.
How can we interact with someone who might be able to communicate that through art?
Yeah, that has been happening, and a lot of my colleagues who are in fields like atmospheric and ocean sciences or neuroscience at the Institute of the Environment or the Institute for Society and Genetics, or in the various fields of biology, they, I think, often want to reach out to writers even more than artists. They want people to tell their story, to help them communicate to a public. And I think particularly in an age of a lot of anti-science rhetoric in the United States and skepticism about the scientific findings that are publicized, there's even more of that kind of drive. I need someone who can tell this story well. And I think there's more uncertainty about whether visual artists have that same capacity. So, does that? Yeah, but there are examples. I mean, a couple of these bioartists, the scientists did reach out to them upon discovering what they were doing. This architect in particular, who I'll mention at the very end if there's time. So let me just offer my sort of what might be kind of provocation here about what I think they're doing around environmental movements broadly, and the sort of the values that have informed environmentalism in the U.S. And what that has to say about the kind of models for food production that they're thinking through and thinking about. So this is a critic who's written about bioart, and he suggests that bioart shifts the locus of environmentalism from conservation of existing ecosystems, of existing species, of existing ecosystem services, toward the generation of new species and new kinds of places. So from conservation to generation. And he describes this generative sense of media. And here media means for bioartists both media in this sense of the sort of visual and sonic and verbal media that the arts rely on, but also media in the biological sense of medium, of nutritive medium, and so on. So what I want to do in talking through a couple of the artists that I find most compelling and who are dealing most squarely with agriculture and with food is to first give you just a really brief snippet from a recent article in which I try to flesh out what they have to say about environmentalism and about sustainable food systems in particular. This is just a really short snippet. From the artist collective Critical Art Ensemble's Free Range Grain Project, which is a mobile lab for testing foods from your pantry for traces of GMOs, to Oran Katz and Ayinat Zur's Disembodied Cuisine, which uses techniques of tissue culturing to create meat in vitro, and then stages a kind of dinner party in the gallery where we eat it, sort of like this Iron Chef contest. So from these two examples, we see that bioartists are mixing artistic and scientific experimentation with an ethic of amateurism. The amateurism, the spirit of amateurism that they see themselves sharing with home chefs and backyard gardeners. Culturing foods in lab settings and then bringing these novelties into the public spaces of galleries and cooking demonstrations. These projects question the boundary between synthetic and organic and between animal and plant. These are also experiments in reculturing. So they're culturing in the biological sense, like they're actually often literally culturing, working with cell cultures. But they're also culturing in the other sense of the word, asking us to sort of rethink where and how and what we eat. And here they are really putting into practice something that the political theorist Jane Bennett describes as the hustle and flow of bodies that constitutes any food system. She says that we have to remember we are always both eater and eaten. And a lot of the new research coming out of the microbial field suggests absolutely we are also food source. We are a kind of incredible kitchen for the microbes within us. And so she says these boundaries we have that we imagine where we are the producer and the consumer and in control. A lot of new research and thinking is questioning that, and I think these bioartists also. So in this vein, those bioartists who culture food are taking up a key premise of agribusiness, that profit inheres in intellectual property and hence in demarcating the boundaries between genes, organisms, and between each manufactured product. So this kind of paradigm in agribusiness, the reason taxonomy matters so much is because it's through these sort of boundaries that you say that Monsanto's BT corn is not the same as, right, something else and is worth more because of it. So these boundaries are what make food today both commodifiable and profitable. The art science labs and bioart groups that I've been sort of researching are disrupting these as much as anything, I think. And in this process, this is where I think they're offering us a new kind of environmentalism. And it's one really informed, as I'll say a lot more tonight, by digital culture, by an idea of the open source and the sort of open access knowledge framework. Okay, so I've offered you a couple things. One, that there's a sense of generating instead of conserving resources, and two, that they're really invested in not the commodity, but rather the sort of open source. So let me show you a couple more examples then. This is the Urban Space Station. Natalie Jeremijinko is a pretty well-known New York artist. She's absolutely brilliant, absolutely crazy. I would say that to her face. But she's wonderful. And she is, I don't know how she does all the different things she does. So she does both prototyping and performance art, and she collaborates extensively with scientists. And she has this group called the Environmental Health Clinic in New York under NYU. And it is a kind of laboratory space where all different kinds of folks meet to explore how to address some of our most pressing environmental crises, particularly those that are urban in nature. So for example, away from the food topic for a minute, she did this project to bring more attention in New York to the air quality and how severe air pollution in New York City is. And so she had a team of about 100 people that she commissioned to, for 2 days, walk around New York with the white medical mask and the symbol of her environmental health clinic, which is the medical symbol turned on its side. And they got dirtier and dirtier and dirtier. And I think they were wearing T-shirts that sort of said something, some sort of word about the project that directed people to maybe the project site. And these were all videotaped. And this actually generated a lot of attention and also debate about, oh, wow, like, and of course, right, of course, scientists have long known, right, that New York City, like Los Angeles, where I live, are two of the most polluted cities in terms of air quality. And it stands to reason. But to have this sort of very visceral, experiential way of communicating that that information proved very powerful. So increasingly what she's doing, though, is prototyping in the same way that Silicon Valley prototypes all of its products. But she's prototyping with not commodification in mind, but with social change in mind. So one of these is about agriculture and the other is about eating. The urban space station, as you can see, is a sort of prototype for doing very high-yield, intensive, biodiverse agriculture in extremely small amount of space. And so that's one of the designs for it. She has a related project called the Ag Bag. And the Ag Bag is that material that the post office uses to send, it's like a Kevlar, is that right? It's like a space-age material. And it proves to be a great material for growing vegetables in. And these can hang off any windowsill in New York City, for example. So she's been distributing these with her same sort of symbol and trying to really participate this way in the growing urban agriculture movement in New York City. So that's on the agriculture side. And then on the eating side is her cross-species adventure club. And so this sort of takes us back to the Iron Chef example, but instead of in vitro meat, instead of a kind of transgenic, or I should just say biotechnological input, she instead draws ingredients from the non-human diet of the immediate ecosystem. So the fungi and grasses that fowl and amphibian are eating in the Hudson River, for example, and then integrates these non-human ingredients into molecular gastronomy meals. And so it's this sort of mixing of an incredibly ecocentric framework for eating with this incredibly high-tech and elite style of cooking. And she has these sort of dinner parties. It's a kind of supper club, really, where each time the wild ingredient on the menu comes from the immediate environment. So they're also beautiful, these dishes, like much molecular gastronomy. And so here again, we see this sort of muddying of the boundary between technology and ecology. But also, I think, in Jaramidjenko's case, she wants us to think about how we can imagine ourselves as being part of the ecosystem at the same time that we embrace our sort of species trait of being technological makers, of being driven towards technology. So it's for her, rather than the anti-technology of sort of bias of 1960s environmentalism, it's this complex dance between a sort of embrace of innovation and technology and an embrace of an incredibly ecocentric way of being in the world. And that's what I find really smart about this. You know, it's suggesting that the way forward is not either to let Microsoft and Intel and Google tell us how to farm and what to eat and count our calories, or to reject out of hand all forms of technological innovation. It's some very complex marriage between those. And then, let's see, this I'll just pause on because it's a really interesting case of some of the other kinds of conflicts that come up when artists and scientists collaborate. There's the issue that I think Louisa raised about, you know, what about the just fundamental differences between what counts as knowledge and what the methodologies are, and, you know, the risk of making that murky. But there are also legal challenges that speak volumes about how we define these two different realms. So this is free-range grain. Has anyone heard about this project or about the artist Steve Kurtz? It made headlines, Colin? Only because you read my stuff. So Critical Art Ensemble, very radical group. Not less in terms of the sort of visual aspect of their art and more in terms of its politics. So they are a kind of anonymous collective, and they are extremely critical of genetically modified organisms and of transgenic seeds and of the patenting of those things. So they did this project back in 2004 called Free Range Grain that I mentioned a minute ago, where it was a kind of mobile, rudimentary molecular biology lab, and it was set up so that, and they worked with a scientist at, I think it was at Wellesley I can't remember now the institution. They worked with a scientist and his group to set this up. And it was really, as you can see, really rudimentary. But in short, people could bring foods from their pantries in and have a basic test run to say if there were any trace elements of GMOs. And it really wasn't meant to give a lot of information. It was meant to make visible that actually these have become pervasive in our food supply. And they started the project in Europe. So they went to several locations in Europe. This was right around the time that there was the banning of importing genetically modified foods from the US. And it, of course, landed the EU in a big case at the World Trade Organization that the United States filed an anti-free trade suit. So in any case, that was the political context. And so to make visible to European consumers that, in fact, it's too late. They are here, right? And also to sort of remind us that so much of the edible world is invisible to us, right? And actually requires the techniques and the methodologies of science for us to navigate and understand. They moved the exhibit over to the US. And this is the image from Mass MoCA, a really famous experimental contemporary art museum near Williams College in Western Mass. And it was at this point, even though the main artist, Steve Kurtz, is American and had been working on this ahead of time, was at this point they suddenly wound up in a bunch of legal trouble. Steve Kurtz's wife happened to pass away of cardiac failure. And when the coroner was there, the coroner reported to the FBI that he had seen these strange things like vials and stuff in this guy's garage. And because he was an artist, the coroner knew, he thought this was odd. So the FBI came and investigated. And in short, the FBI ended up arresting Kurtz on charges of bioterrorism for the tissue and cell samples that were in his garage, which he had gotten in the mail from his scientist collaborator, the one whose institution I can't remember. So eventually the bioterrorism charge was knocked down to mail fraud, but this dragged on for almost 3 years in court. And finally, because there was a huge outpouring of support for Kurtz, he was vindicated. But the mail fraud case the mail fraud charge is what I wanted to pause on. That had legs for the federal prosecutor only because of our sort of current regulatory apparatus, which says, as many of you may know, that only scientists registered with the ATCC can ship biological samples to one another. So only scientists can ship these biological objects, if you will, across state lines, or probably anywhere, in fact, not just state lines. Is it state lines, or is it any form of shipping? Yeah. And so I pause on that because it suggests that we have this very evolved bureaucracy that says that there are scientists and there are non-scientists. And we actually have created a kind of very real barrier to collaboration. And the reasons for that are sound. There are many good reasons why not just anyone should put biological samples in the mail, arguably. But it did expose this kind of, this sort of problem for artists trying to do really sustained, deep collaboration with scientists. So I wanted to highlight that. And this is just a bit about the sort of context for the trial. So the prosecutor said this was informal exchange. It was, again, it was the work of amateur and thus a problem from a legal standpoint. Okay, and then I'm just seeing the time. I'm gonna stop. I think I'm just gonna go forward to one other example. Just bear with me. I want to get to it. Okay, this last example, and then I'll end there. This is the other group that I mentioned, and they, Oren Katz and Einat Zur, are at this this Institute for Wet Biology Art in Western Australia. And they have a project called Disembodied Cuisine, which is another in vitro meat project. But it actually began in the sciences. It began when they were funded by Harvard Medical School to culture natal sheep muscle cell onto a polymer as an in vitro meat experiment. And then it morphed into an art project. So I wanted to just highlight an example that went the sort of other way. And it morphed, in particular, into a very provocative supper club. So this is, like Natalie Jeremchenko, they held a dinner party in a gallery as the final stage in their in vitro meat experiment, except they really highlighted the risks, right? The dangers, both imagined and real, of breaking our sort of usual paradigm for food. And I thought that I just ended here because I think it's not all rosy, it's not all utopian in the world of bioart. Many of them are actually provoking us to ask about the risks that our technological innovations and inventions may be posing. And because in the second stage of the Disembodied Cuisine project, the tissue culture was grown from frog cells, in these aquariums you see to the left there of the hermetically sealed biohazard dining room, makes me think of something Stanley Kubrick would have done. So these were the frogs that from whom the cell samples were taken, the tissue samples were taken. So they sat there and stared at you throughout the meal, and then they were released into a local park, which I always thought maybe that wasn't such a good idea on the part of the artists. I mean, I'm not sure if they got clearance on that from that standpoint. So. Yeah, I'm not sure if this is an invasive species of frog. I mean, anyways. And these are just some shots from their lab of the actual centrifugal sort of setup. So, so I'll just end with their quote. I didn't get to the meat house. Maybe I'll end up showing that tonight. So Zur says about this project, which again they call it a semi-living steak, which I find a really interesting framework. On the ethical level, the project addresses the most common zone of interaction between humans and the living world, and also probes the apparent uneasiness people feel when someone messes with their food. The project offers a form of victimless meat consumption, but you can see that he himself puts victimless in quotation marks, right, to sort of call attention to the fact that whether even test tube food can ever truly be victimless. And then, I'm just going to go a little forward here. I just wanted to end with this one slide. So I had some things to say about slow food, but that's going to be at the heart of tonight, so I won't dwell on that now. But I gave a sort of preview of my argument that there's new values for environmentalism, and I thought it would be helpful to end with this last slide. So on the left are, to my mind, and I would be really curious what you would add to both sides of this equation, to my mind are three of the organizing values for environmentalism and for sustainability that took shape in the United States, especially in the 1960s, and then gained steam over the last 50 years, and that are particularly important both in food sustainability and then also in all sorts of other branches of environmental sustainability. So protection of habitat, conservation of resources, and remediation and restoration. And I also would say that one of the key words is on this side biodiversity as well. But what we see sort of being explored in bioart is whether we now must generate biodiversity rather than conserve it. And I think this is very much in line with this wild new branch of science, de-extinction, that some of you may have been following. These scientists who are thinking about using genetics to bring back to life species that have gone extinct, known as de-extinction. If you haven't been following this at all, it's really fascinating, troubling, interesting area of scientific research right now. So generating biodiversity and then remixing, as I'm using remixing for those of you who are fans of music in the sense of DJs sampling from different genres and different styles, like remixing the ecological and the technological, as we see with Cross Species Adventure Club, really saying to us that environmentalism can't afford to position itself as anti-technological. And then this kind of improvisational, adaptive approach to environmental risks and environmental crises, rather than the sense that we can get back to the prior state. There's this sense of sort of damage or less than perfect circumstances being sort of with us no matter what. So yeah, so I'll end there, and I'd love to take some more questions. I think we have about 10 minutes.